Matthew Diamond, who is based in Trieste in Italy. Matthew has been working, I think, for nearly all his career in the somatosensory system. But what I think is really distinctive about Matthew's approach is that he goes well beyond the transducer, which is in his case the whisker of the rat, and he goes beyond processing in the primary sensory area, which is S1, which we've also heard about quite a lot the last 2 weeks. And he looks further into how the animal is using that information to make decisions. And so Matthew has looked up the pathway higher into cortex, up into places like hippocampus, to see what kind of information is being extracted from those somatosensory signals and how they're guiding the animal in real tasks. And I'm not sure what he'll talk about us today because it's apparently going to be new material. So I'm really looking forward to hearing what he has to say. Matthew.
Unpublished. Is this on? Is this working? Okay, great. Yeah, unpublished material, as Henry points out. So this is our location in Trieste. This is actually a photograph taken from the terrace of my office. And it's the same piece of water that you have here in Barcelona. This is the very farthest north point on the Mediterranean Sea, so we're connected to Barcelona by this body of water. And so for sort of a student audience, I've looked at very quickly at your posters, and I can see that you come from backgrounds that are not strictly neuroscience and not strictly cognitive neuroscience. And so I'll just spend a few words on the strategy that we use in cognitive neuroscience. The cognitive neuroscience of sensation and perception, that's our field. And I explain it to my own students using this triangle, which is a golden triangle both in color and in proportions. I made this using the ratio of that the Greeks considered to be the perfect ratio between the sides of a triangle or the sides of a rectangle. And what we have in the cognitive neuroscience of sensation and perception, well, there's two words: cognition, which means that we're interested in behavior and thinking and decision making, not only in the simplest functions like reflexes, and neuroscience, which means that we're interested in the brain. You could have cognitive science without studying the brain. That's perfectly valid. Or you can have neuroscience without studying cognition, which is perfectly valid. But here we try to put these things together. And the way that we do it is shown in this triangle, where in every experiment we try to have three elements. One is a quantified sensory input, and because we want to know exactly what's coming into the brain, what kind of signal the brain has available in order to make its choice. We have a measure of the human or rat percept, so they have to do something. They have to actually express, they have to show that they are dealing with this input, so we have to have a measure. The animal's not anesthetized. Anesthetized studies can be extremely useful to get an experiment going, but in the final stage we want to actually understand how the animal is using the information. And then neuronal activity. And then from the triangular relationship, we can study what happens between the sensory input and the perceptive behavior. That's the field of neuroscience known as psychophysics. We can explore how a sensory input is converted into neuronal activity. This is sensory coding, and it's extraordinarily important because sensory systems, the different sensory systems integrate completely different kinds of physical energy from the world, going from air vibrations in the auditory system to chemicals in taste and olfaction, skin vibrations, as we'll talk about today. But it all has to get into the same language, into the action potentials that the brain uses to communicate and transmit information. And so coding is the process by which some physical input is converted into the language of the brain, neuronal activity. Then we're also interested in how neuronal activity that represents a stimulus leads to behavior. And this we can call decoding or decision making. So mostly today I'll talk about these two, this level, an input and a behavior, but I'll add some neuronal activity just so we can complete the triangle. And like Henry, I'm also concerned about not getting to the end or forgetting to mention my collaborators, so I put it in the beginning instead of at the traditional end point of the talk. So these are some of the people, not all of them. Arash Fasihi, he's done the lion's share of this work. He's the main figure in most of these experiments. So if I don't mention other people, then the default is that Arash has done the work. Vincent's did some computational work. He's now in a startup technology company in Berlin. Amir is a PhD student. Shima is finishing her PhD. Vahid was a PhD student. He's now at EPFL in Lausanne. And Athena is now at Princeton. I'd also like to mention Francesca Pulecki. She takes care of our rats and trains the animals a lot and also does surgery. She's a critical contributor. And also a group that builds and programs the instrumentation. That group is formed by Fabrizio Manzino, Marco Gigante, Eric Zorzin, and Fabio Meneghini. I don't have photographs of them. So that's the research group. And in the sense of touch in rats, I think that you've heard about this from Tony. I'm not sure exactly from Tony Prescott. I'm not sure exactly the organization of the course. But you've probably heard terms like active sensation, and I've seen also in your posters that a lot of you are interested in the relationship between motor systems and sensory systems. And the whisker system in rodents is really a prime example of active sensing, because the sensory system doesn't just wait for things to happen, but makes things happen by the way that it interacts with the world. It actually creates stimuli. So there's a very strong interplay between the incoming signals, the sensory systems, and the outgoing signals, the motor control. You've probably seen, or you may be aware that rats and mice and other rodents explore the world by moving their whiskers and essentially by creating stimuli by their own actions. But I'd like to highlight today, while I believe very deeply in that form of perception, which I call generative perception, because the animal generates the stimuli by its own actions. I'd like to point out another way that animals may learn about the world, which is active, but we call it receptive sensation, because the energy already exists in the external world, the mechanical energy. So the animal doesn't have to create it because it's already there. And if the energy exists in an external object, then the animal's own actions could actually create confusion. Is it possible not to have this blinding in the booth? Can you hear me? Maybe this is a little bit sort of blinding me. So if the energy exists in objects already in the world, then the animal's own movements might actually create confusion. And in those cases, you might expect the animal to actively suppress its motor output in order to more accurately acquire the signals that exist outside. So that we also think is an active process, and we call it receptive sensing. That's what I'll talk about today. So how might an animal use receptive sensing? So Marco Gigante, one of my technicians, also a good artist, and he drew this rodent in a tunnel, which is their normal. I don't know if you guys are going to figure this out sooner or later, but the rodent is in its normal environment, which is an underground tunnel in the dark. It feels its way around the tunnel, sniffs its way around the tunnel, and at a certain point he wants to come out and get food. It's a moment of danger because when he jumps out of the hole, there may be something there that's waiting to eat him. So this is a fantasy. That's why we don't have photographs or movies. We have a drawing. So according to his fantasy, before he jumps out, he might want to figure out what's in the world. And he could do that from holding the whiskers in contact with the walls and the ground and feeling if there's vibrations. So a vibration that's sort of low in amplitude and decreasing might, for the animal, mean that there's something there, but it's small and it's moving away. So in a moment, it's safe to come out. In contrast, if it's a large vibration and it's getting larger, it means maybe that there's a big guy there. He's getting closer, and it's not the right time to come out of the tunnel. So it's as I said, and I repeat it, it's a fantasy, but I think it's maybe a starting point for thinking about how mice might feel vibrations, why they might want to feel vibrations. So we made in the laboratory an apparatus and a behavioral task where the animal has to do exactly this. They have to measure vibrations and compare them over time. The apparatus looks like this. Oh, sorry, I'm not ready for the apparatus. So the way that the task that we set up is a delayed comparison working memory task, where the animal has to compare pairs of sequentially delivered vibrations. So delayed comparison working memory task is particularly fascinating and important and intriguing for cognitive neuroscientists, because within one trial, and the animal can do 200, 300 trials in a day, within one trial, which lasts in the order of 3, 5, 10 seconds, there's a sequence of cognitive operations which go from preparing for the trial, which involves attention, encoding the first stimulus, S1, holding the memory of the first stimulus, encoding the second, comparing to the first to the second, and making a choice. So within this, there's a lot of interesting operations, which any one of which, how does the brain encode a stimulus? That by itself is a great question. How does it hold a memory? That's equally important. And yet, within 5 seconds, you have all of this. And you can design the task in a way that you can separate these different operations. So for a cognitive neuroscientist, it's a behavioral task. It's a structure that is very rich and holds a lot of possible observations for us. So the way that we do this in laboratories, we have a big motor which gives us a lot of flexibility that creates a vibration, and the rat learns to keep its nose in a little tiny nose poke, by which its whiskers rest on a plate, and the plate is controlled by a motor. And the rat receives vibrations, and at the end of the second vibration, it has to make a choice either to go left or right. And if it makes the correct choice, according to the relationship between the two stimuli, then the rat gets a reward. If it makes the wrong choice, it doesn't get a reward. There's never any punishment. The only punishment is the failure to get the reward and the need to wait for the next trial then. And what it actually looks like is this. This is a box built by the technical group that I mentioned before in plexiglass, and this is the motor, this is the plate, and from inside it looks like this. So the rat's point of view, he puts his head through here. This is a nose poke, and there's an optic sensor there, and he has to keep the tip of his nose exactly in that hole for the entire trial. If he leaves before he gets the go cue, then that trial is aborted and he has to wait for another trial. So he's almost motionless inside here. The whiskers lie on this plate, and the plate is controlled by the motor. So this is how we stimulate the whiskers. And at the end of the second stimulus with the go cue, the rat takes his head back out and goes either to the left or to the right and hopes to get a reward. And of course, you can ask questions if they come up during this presentation. This is the structure of a trial. There's the pre-stimulus delay when the rat focuses its attention, and maybe I'll touch on that if we have time. It receives the first vibration, then there's a delay between stimuli, a second vibration, post-stimulus delay, he has to stay in the nose poke, and then a go cue. And what's fun for the research team is that any possible variable within this we can control by the computer. We decide which are the experimental variables. We can control, for example, the duration of the first stimulus, duration of the second stimulus, that's the main topic today. The intensities of the stimuli. So these two things together, the intensity or the amplitude of the vibration and the duration are the features that I'll talk about today. But other things are also interesting. For example, the interstimulus delay. How does behavior and how does neuronal activity vary according to how long the animal has to store the information in memory, right? Because the load on memory increases if you have to remember the stimulus for longer. So that's another interesting thing that we play with, but it's not the topic of today. Okay, so unfortunately, I collect slides from the last couple of years, and sometimes we change terminology. So I'm a bit sloppy in terminology here, and I apologize for that. So the first vibration we sometimes call S1 for stimulus 1, vibration 1, V1 for vibration 1, and it's characterized by an intensity, which I'll explain in a few minutes, which we sometimes call sigma base or sometimes sigma 1, and then the parallel terminology for the second vibration. And I apologize that in different figures you'll see different terms here. I just haven't cleaned that all up yet. Okay, so now this is the first thing that has to be attended to, the nature of the vibration. And we make the vibration by beginning with a normal distribution, a perfect Gaussian distribution, centered on 0 in velocity, in the velocity dimension. So they're positive and negative velocities. And the way we make a vibration is in each time step, sampling a value from this normal distribution according to the probability distribution function, and then stringing those samples together, and then applying a filter. So once was that a question or a stretch? Okay, stretch. Okay, so so when you sample the velocity values, string them together, you get a vibration. And by the sort of inverse, since you're sampling from here, once you get the vibration, the statistical structure of this vibration will reflect the distribution it was taken from. So this vibration in velocity space has, if you were to project all these values back, the standard deviation is sigma, the same as the distribution it's taken from. And that's the main thing we can play with. We can begin with a wider normal distribution with a larger sigma, and then we get a bigger vibration in the velocity dimension. So now the blue one compared to the red one has more of these sort of large positive and large negative values. And that's what the human or the rat is trained to feel. And this is one trial, and what you'll see is the rat, it's frozen in time as he's finishing collecting the reward from the previous trial, and you'll see that he puts his head here, this vibrates, and he receives the vibration through this motor. He waits. The delay is, I don't know, 1 second, maybe 2 seconds, I'm not sure. Then he gets the second vibration. Then there's a go cue, and he quickly goes to the left. Here's the first vibration. Here's the delay between. Second vibration. He felt the second one, compared them, and turned left. Any questions? Okay, so this is the kind of thing that what we ask the animal to compare, the human to compare, are these two vibrations characterized by their two standard deviations. And it's a parametric task because the thing that has to be compared is a parameter. It's a value along a dimension, sigma. And it's important to understand the difference between a parametric comparison and a categorical comparison. They're conceptually two different things. There's lots of working memory tasks in which animals compare, or humans, whatever, monkeys, compare two objects of a different category. In olfaction, for instance, rats have very good working memory capacities, but we have avoided that kind of a comparison because if the rat is required to compare, let's say, cheese versus sausage and say they're the same or different, versus our experiment where they compare the two different statistical structures, the way the brain deals with this is very different. And our aim was to do the parametric because in the case of two very different objects, we really don't know which neurons encode the two different objects, and we don't know where to find the neurons, and we don't know where to find the neurons that do the comparison. Because there may be a set of neurons, the way the olfactory system is built up, there may be a set of neurons that are involved with encoding the sausage and a set of neurons that are involved with encoding the cheese, and we don't know where the neurons that encode both, which what you'd expect to do the comparison, some neurons have to reflect the convergence of the two representations. We don't know where to find them. We don't know what this code looks like. We don't know what this code looks like. We don't know what the comparison looks like. And so our argument is that you begin quite lost. You can't answer the kinds of questions we want to answer in a categorical task. In contrast, in a parametric task, there's no reason, from what we know about the tactile system, there's no reason to think that a neuron is specialized for a given value of sigma. There's lots of evidence about that. That's not, neurons don't have sigma-specific, that is, velocity-specific receptive fields. Neurons encode velocity in a graded manner. Therefore, the same neurons encode these two different vibrations, and we can find neurons that encode both, which is important, and we can find neurons that do the comparison because they receive both inputs. So that's the design of the study. And as I mentioned, we can do the same thing in humans. As Henry said, it's in a way sort of the golden standard that people want to compare their studies to, because if we say rats can do this well, then it's natural for someone to say, okay, but how well? And as humans, we think that the measure that should be applied is humans. So we can do the exact same experiment in humans, where instead of we don't use our whiskers very much, but we use our fingertips, and so the vibration is applied to the fingertip. Exact same task. And I knew that Professor Ranulfo Romo is on the program for this meeting, but I didn't think he'd be here today, and I didn't. So I actually didn't put in these slides because he's here. I do this in all of my talks, and it's probably, I'm not trying to flatter him, it's probably embarrassing for him. But the work that we do in my laboratory is Professor Romo is the leader in the field, the pioneer in the field, and when we do something, we usually realize that it's already been done. We go back 4 years or 3 years and we say, oh yeah, there was that paper. We do it in a slightly different way, but most of it has been done. So this is a monkey version of the task that's already been done, and the monkey receives vibrations on its finger, and the monkey has to compare the first vibration to the second vibration with a delay between them. And the first vibration is characterized not by the sigma that we use, but by the frequency F1, the second by F2, so the monkey has to do an F1 versus F2. So most of what I'm saying is already known, but we've done some variations in the task, and the main thing that I'll talk about is really the difference, the different computation that has to be done by the brain if it's doing this stochastic noisy vibration that we call the sigma comparison with respect to a sequence of pulses. So of course we want to measure behavior, so there has to be a rule. So we say if sigma base is larger than sigma comparison, then the rat should do one thing, and if the relationship is the opposite, it should do the opposite thing. And so by the action that the animal takes, turning left or right, after a period of a couple of months of training, on any given trial, we have a window into knowing what the animal felt. And we organize the stimuli in a very particular manner. It's important and it's necessary to do this. So we can take 2 stimuli. Every trial has 2 stimuli, right? So we can consider a single trial to have a combination of a base stimulus and a comparison stimulus, or vibration 1 versus vibration 2. So each box could contain an entire trial. And we need to use stimuli, we need to train the animal and test the animal with stimuli from this entire range. There's a decision boundary, because on one side of this diagonal line, the first stimulus is larger than the second, so the rat should do one thing. And on the other side, the relationship is the opposite. The second is larger than the first, so the rat should do the opposite thing. So this is a line that separates the 2 actions that the animal or the human, by pressing a button, not by turning left or right, but by pressing one button or the other. This diagonal separates the 2 choices that should be made. So why do we need this structure? Because humans, and especially rats, probably monkeys as well, would like to do a task as easily as possible, as simply as possible, because the brain wants to be efficient and economical. And if there's and working memory is hard. It's a big cognitive load. If you're ever a subject in our experiment, come by Trieste, we can test you. And you see that it takes a lot of attention, it takes a lot of effort to put things in memory. Especially, difficult things where, it's not like, I'm going to give you a word. A word is easy to remember. This is hard to remember. And so it would be nice to have a shortcut, the shortcut being to attend to one of the 2 stimuli and not to the other, because one stimulus is easier to process than 2. But here it's impossible to take that shortcut, because any given value of a first stimulus can be followed by 2 different second stimuli. And the values of the second stimulus determine the choice that has to be made. So a subject can't just attend to the first stimulus, because if they attend just to the first stimulus and try to make a choice, there'll be a chance, because it's the relation of the second to the first that determines the correct action. And they can't attend only to the second, because a given value for the second stimulus can be preceded by 2 values of the first stimulus, and the relationship again determines what is the correct choice. So if you try to do this just by attending to the second stimulus, make some threshold, like if it's big, turn right, if it's small, turn left, using just one stimulus, you'll fail. Well, you won't fail, you'll be at chance, or close to chance. And to do the task with good performance, 70, 80, 90%, you need to use both stimuli. So this design forces the human or rat to do a working memory task. Yes?
Is that also true for the pairs of stimuli on the extremes of this sort of rectangle?
No, on the extremes, if the subject is clever enough to detect that this is an extreme first stimulus, then it could actually ignore the second stimulus and it would know the answer. Or if they felt that this is the weakest first stimulus, they could ignore the second. But there's boatloads of data showing that that's not what people do. And in fact, they make a lot of mistakes on exactly this stimulus, fewer mistakes on this, but a significant number on this. And we know why. It's because they're using something called contraction bias. But the bottom line is that they don't actually treat this stimulus as being different from the other, or this. They don't try to simplify it for that. Okay, so then we also want to measure acuity. This is a design that requires subjects to do a working memory task, but doesn't give you a very good measure of acuity, of how sharp the system is. And to get the acuity, we fill in these bars with lots of stimulus pairs that are very closely spaced, and then seeing how performance changes according to very small differences in the relationship between first and second, we can get measures of acuity. So the first data I'll show you are based on experiments with 400 milliseconds, 800 milliseconds, 400 milliseconds. And psychometric curve, I want to find out who knows this. Raise your hand if you know what a psychometric curve is. Okay, so raise your hand if you don't know what a psychometric curve is. Everyone should have raised their hand for the first or the second question. Okay, so enough people don't know that I'll explain it. So a psychometric curve is a very, very simple measurement used in psychophysics that allows you, with a very easy computation, to know a lot about the way the stimulus is sensed and perceived. And all it does is on one axis give the stimulus value, and on the other axis the choice of the subject on average across many trials. So for us, the stimulus value is actually a difference between the stimuli. So it's sigma 2 minus sigma 1, not just a single stimulus, but both stimuli together, normalized to their sum. So for us, the positive values mean that sigma 2 is larger than sigma 1. So one choice should be made. For negative values, it means that sigma 1 is larger than sigma 2, and the opposite choice should be made. And then this axis measures the percent of the time that the subject actually makes those choices. So a perfect subject, what is sometimes called an ideal observer, would look like this, because when sigma 2 minus sigma 1 is positive, sigma 2 is larger than sigma 1. So for 100% of those trials, the subject should say it's larger. And when sigma 1 is larger than sigma 2, which means these values of the stimulus pair, the subject should, for 0% of trials, say sigma 2 is larger than sigma 1. So you get a step function. I don't know exactly. Yeah, this would be 50%. So you have a step function for a perfect observer. But so that's what you actually compare performance to. That's the perfect performance. But nobody is perfect, no human or no rat or no monkey. And so the curve never looks like a step function. What sort of above chance but not good performance might look like is something like this. The curves always have a sort of an S shape. Here the S is stretched, so it's called sigmoid sometimes. And for someone who's doing the task but is not an outstanding performer, the sigmoid would be rather shallow, and as it gets steeper, performance is better and better. So this last one here is, it's kind of getting closer to the step function. So that means that a subject with this shape of psychometric curve is doing better. And so what I show you now is the psychometric curves with the same scale for 6 rats with the average in red, and for 18 humans with the average in blue. And you can see that the humans get to almost 100% for the easier stimuli, the rat don't. And you can see that there's this steepness of the psychometric curve, which is better for humans. So if we take, but there's some variability in rats and some variability in humans. So if we characterize each subject by 2 measures, the percent correct overall stimuli and the steepness of the psychometric curve, then we can plot all the subjects in a scatter plot. And this is percent correct, this is the slope. Blue are the humans with the average here, red are the rats with the average here. You can see on average the humans are better in percent correct and in steepness of the curve than the rats. But very good rats, like these 2 here, this one is quite good, are better than less good humans. So these are the cognitive neuroscience students, and these are the theoretical physics students, and these are the mathematics students. Because most of our subjects are PhD students from the university, and we have 3 degrees, neuroscience, physics, and math. And the math students just don't get it. Yeah.
What were the stimuli?
The stimuli are the vibrations.
Vibrations,
Yeah.
sound.
No vibrations. So you're asking, do the vibrations make sounds?
Yes, depends on the zone where you...
Yeah. So the humans,
Because you have this problem of masking.
yeah, humans can hear the vibrations, but they have earphones, and so they can't hear them. We do control tests that,
Okay, you impose a constraint.
yeah, we do controls that if they can hear it, but if they have the sound available but can't feel it, then they're a chance. So the ear plugs work. In rats, we do the same thing in a different way. We allow them to hear the stimuli without feeling it, and their performance goes to chance, which confirms what we did acoustic measurements of the stimuli, and the stimuli that we use produce lower frequency sounds, which are below what is believed to be from the literature, it's below the hearing range for rats. So they shouldn't be able to hear it, but you can't really believe the literature, so we actually did the control and they can't hear it. So they're actually doing it by touch.
But now, Matthew, I assume you're not stimulating the humans via some facial hair. So
No, fingertips.
exactly. So there might be a difference there in sensitivity between whisker system and fingertips that might
Yeah.
account for the difference you see.
Yeah.
This is also what you believe, or you think it's really that the humans that are better understanding in picking up the rule of the decision making?
It's
Or
it's
both?
it's a combination of both.
Okay.
The evidence that humans are better with the rule is that rats make errors 10 or 15% of the time, even for easy stimuli, and their sensory system should not be making that mistake. And the reason that they make mistakes on easy trials 10 or 15% of the time is something called lapse rate, as you know, which means errors based not on sensitivity, but based on either not understanding the rule or forgetting the rule, or there's something also that people think that rats do. There's good reason to think that rats do this, which is they test the opposite rule. So they make errors because they want to be more than humans. Humans and monkeys, if they think they know the rule, they don't care. They don't want to test that the possibility that the rule has changed. Whereas rats, even if they think they're going to get a reward on one side, they just want to be sure that they're not going to get the reward on the other side. This is called the matching rule. It's been studied by psychologists for 60, 70 years. So rats make errors, which are not really errors, it's testing of the world.
So do you agree with the theory of regret?
Of what?
Regret. The rats are able to regret a choice.
Like
Since
Dave
we-
Radish's
Yeah,
experiment.
the Reddish experiment.
I think that they do, yeah. I think that they do, and we're kind of interested in, we have some experiments that are going into things like, I'd say not regret, but confidence and certainty and things like that. And if you probe them, you can find that they actually have these workings underneath the choice about certainty, expectation, confidence, and so on.
So would you predict that if they, in some trials you give them more reward and they know that this trial will have a higher reward,
How do they know it before?
I don't know, with a cue, saying that
A cue that says it's a high reward trial, then what?
they will then drop this error in very easy trials.
I don't know, but I guess my prediction would be that if you have a cue that says this is a high reward trial, they would probably do what they think the rule, what the rule tells them to do. So they would probably not do this exploration of the possibility of a different rule on the high reward trials. That would be my guess.
Can I just quickly ask about whether or not there's an active component to their performance improvement? So their ability to move their whiskers, for example. I don't know if there's any data on that. Maybe they increasingly fine-tune the motor control of their whisker movements
Yeah.
so that they can better pick up the vibrations.
We haven't quantified that, but we've looked at it a lot, and because it's very hard to collect the data in such a way that you can prove this, but our very strong impression, going back to what is active sensing and what is generative and receptive, is that the better trained they are, the more they suppress the whisking during the trial. So we see them, we make high-speed video, and we see them going into the nose poke, whisking a lot, because it's in the dark, and they're navigating through the world through active whisking, because they have to find this hole, they have to find the nose poke. So they're doing a lot of movement to find that. Then when they get in the right position, during this pre-stimulus delay, the whisking really just goes down almost to zero. And then interestingly, in the delay between trials, they do move a little bit, and then they suppress again. So we think that there's, and the good rats in general are really good at this suppression. So we think that actually being able to go into this receptive mode is correlated with good performance. It's not the only thing, but it's part of it.
Is there some literature on the kind of the bandwidth of frequency that rats can feel through the whiskers?
Yeah.
What is the upper limit?
So neurons, so the bandwidth is usually, it's better to think of it not in frequency but as in the speed, because high frequency means high speed. And what they actually feel, what causes the neuronal firing, is the speed of movement, which you can consider in angular change at the base of the whisker. And they have, so they show again sigmoid functions. There can be speeds that are below the threshold for causing neuronal firing, then the firing goes up and then it saturates. And this, our stimuli are sort of covering, the weakest stimuli are above the threshold, so we do produce activity, otherwise the rats couldn't do the task. And the strongest are still below the saturation for most neurons.
One more question here.
Yeah.
So the way you have defined your stimulus on the x-axis
Yeah.
is sort of the relative difference of the two signals.
For the psychometric
Did you
curve.
test that that's in fact the case, that they are sensitive to the relative difference and not the absolute? Like, that didn't have to be the case, right? That relative, I'm talking about that you're
Yeah,
dividing
we
by
make,
sigma 1 plus sigma 2.
yeah, we can do that, and by making this. Okay, so here oops. So here we make the psychometric curve here, right? And here we average the two. So this and this are inside here. So we can move this down to this place, and the psychometric curves are the same. But they have to adapt to the range. So if you give a lot of stimuli up here, and then very few stimuli here, it won't be as good, because the performance gets better within the range that's given more commonly. So the answer is yes, they can get just as good a psychometric curve here as here. The psychometric curve doesn't change, provided that you give the same proportion of stimuli in that range. Okay, so onward. Okay, so humans are on average better than rats, but it's sort of comparable, which implies that somehow the brain is able, very different brains are able to accomplish this task in not a completely different way. Sorry for the double negative. So back to the experiments that we're kind of replicating in the rats. We have monkeys doing a frequency comparison, but I want to point out the main difference between the structure of our experiment and those of the Romo group, namely that if the subject is comparing a train of pulses or a sinusoidal frequency, both have been used in different studies, and if these are periodic, then the information that's available to the brain, to the subject, can actually be collected in a very, very short period of time. So go back again to the concept of the ideal observer as the perfect computationally errorless system. If you have to judge frequency, how much do you need here? You need one pulse, one interval, and then a second pulse. And that's frequency, 1 over period. That's all you need. But, of course, the train goes for 15 or 20 or 30 pulses, and performance gets better when the train lasts longer. But it's not because there's new information. It's the same thing presented over and over again. And again, for sinusoidal stimuli, if you're judging frequency, then for the perfect observer, you need one cycle. We get better when we have more, but it's because the brain can confirm and reestablish the same information. And that's different from the stochastic stimulus that that we use, because even an ideal observer cannot reliably, with 100% certainty, know the stimulus in a very short period of time. It's stochastic. The stimuli come from distributions, and the distributions cannot mathematically be defined instantaneously. So I can illustrate that very simply by starting with this red normal distribution, which is what we draw samples from. So this is the beginning distribution with some standard deviation. And I've played the vibration for about 30 milliseconds and taken out these samples from it. So if you have to do a sigma comparison, then you'd need to know, you have a 30 millisecond vibration, you have to estimate sigma of that vibration based on these samples. You'd have a hard time saying that you know the shape of the red distribution from the samples that you have at that point, right? You just don't have enough samples. And you know this in your very beginning statistical course, that you can make better comparisons, more reliable comparisons when you have more samples. So I'll start this movie, and what you see is progressively more samples. The vibration is played out in time. You'll see more samples, and what you'll see is that from about 100, 200 milliseconds onwards, the distribution is well represented by the samples that came from it. So now you could say, okay, I think I know what this distribution is. So from 100 to 200 it gets better. Now it's at 400, it's still better. And then your visual system tells you that from 400 onwards it's not getting that much better. So you more or less have most of the information that you could get within about 300-400 milliseconds. And so you collect, so the samples that provide you the necessary information first accumulate very rapidly and then begin to saturate. That's just statistics, nothing neuroscience about that. So that's different from the repeated stimulus, and it's an observation that's been made in motion perception in humans and monkeys in very, 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 very famous experiments from Newsom and Shadlin and many of their collaborators, where there are these dots that move on the screen, and they can be either completely uncorrelated with each other. So the task is to detect the direction of the dots, the global direction, the overall direction. And if they're completely uncorrelated, there's no right answer. They're all moving in an unrelated way. If they're 100% correlated, then it's very easy. But if they're 10%, 20%, 30% correlated, then it becomes a difficult task. And they've studied this. They've studied the brain areas that are involved in this. And what determines performance is the percent of coherence and also the time that's available to do it. So if there's a very low degree of coherence, then the subject needs more time to actually get the necessary information. If there's a high degree of coherence, the subject needs less time. So these are the kinds of things that we play with in the vibration. Coherence is stimulus difference, and time is time. So let me rephrase this question. For an ideal observer and accumulator, accuracy increases with the number of samples. Can rats accumulate over time, and do they do it in a way that's similar to humans? The answer is that they can, and it's an interesting answer because it's not, how would you say it? It's not, it doesn't have to be this way. It's possible that rats, it could be that rats don't accumulate evidence over time. And the reason I say that is because a colleague in the field at Tübingen, Cornelius Schwartz, states very clearly that rats do not accumulate tactile information over time. When they have to make a choice, they make a choice based on the very last thing that's happened, what you could call recency. The last thing that happens before the decision time determines their choice, and what happened previously has no bearing on their choice. They don't accumulate, they don't get better with a longer stimulus. All that they use is the very end of the stimulus, the last 20 or 30 milliseconds. And he says that himself. I'm not distorting his point of view. And what I'll show you is that maybe that's true in his task. In this task, when rats do have a reason to accumulate, they do accumulate in a similar way to humans.
But Matthew, question, the comparison with the motion display.
Yeah.
I think it illustrates the point, but of course there's an important difference as well, which now we have to integrate over time.
Yeah.
In the motion display, it's instantaneous. Right, so I'm looking at the motion display and I can extract instantaneously the information and enforce my decision. Well, in your task, I extract first one chunk, I have to hold it in memory, wait a little bit.
No, but I'm thinking about an individual stimulus, the information that's in one stimulus.
Right, okay.
Yeah.
Okay.
Yeah. So, so Paul raised a good point, and let me clear that up in case this creates some confusion. When I'm talking about accumulating evidence, I'm talking about the evidence within one vibration. But then the rat has to do the same darn thing for the other vibration, and then compare. So it's a really hard task. Okay, so what's our first model for how evidence might be accumulated? And it turns out that it's not right. And that's really what's fun in neuroscience, is that you start out with some mental image of how things are going to work, and you collect, you run experiments, and then it kind of doesn't fit, and then things get interesting. So this is what we thought, and it's not right. So we thought that the subject is essentially taking a mean value of the distribution as the vibration goes in time, and as the stimulus continues in time, the mean essentially gets tighter and tighter. It gets closer and closer, as I showed you in the simulation, to the expected value for that distribution. So this is an orange vibration and a green vibration, and the dark line is one trial, and what's shown here is the nominal value of the sigma, of the standard deviation of the distribution, is right here in the middle and right here in the middle. And the dark one is what happens in one trial if you accumulate values and keep them perfectly in memory, store them, and then at each instant you say, what is the sigma based on all the preceding values in this vibration? So this is one vibration, and it wanders around, so you can see that for a very short vibration, the orange one or the green one, you'd be likely to be pretty far off in your estimate with a very small number of samples. But as the vibration continues, the estimate that's available to the observer gets closer and closer to the expected value. And the shaded line is one standard deviation for thousands of repetitions of different vibrations. This is the shading is the distribution, one standard deviation around the value, with around one standard deviation in the spread of estimated sigma values. So this says in one trial, and then the shading is across many trials, how much variability you'd see in sigma over time. And what you see here is that the shading, again, there's no neuroscience here, it's just a statistical simulation. The shade, which you could also derive analytically, the shading gets narrower, which means essentially that the observer can make a better estimate of sigma. So if the brain is doing this, then after a 200 millisecond vibration, you'd, on average, across many trials, on average you could make the distinction, but this overlap zone means that errors that the brain might make just based on the input that it's received. Because if the green one is here and the red one is here, the subject, even a perfect subject, would make a mistake. If the stimuli last longer, then the overlap, which is the possible error for the perfect observer, it decreases. So errors would be less likely just based on the statistical evidence that's available. Okay? And this is what we see in humans and rats for what we call balanced trials, where the first and second durations are equal to each other but vary from trial to trial. And this is 100, 200, 400, 600. And you can see that for humans and for rats, and it's not perfect because that's I mean, you can never get enough data to be perfect, but the very clear trend is for humans and rats, when the vibration duration goes from 100 to 200 and 200 to 400, the psychometric curves get better. Then from 400 to 600, there's a small difference in the green to red. If by statistical tests you can show that it's a significant difference, but it's quite small. Also in rats, they improve from black to blue and blue to green, and then a little bit also from green to red, but not very much. So this evidence is consistent with the model that I first presented. So, okay, we're fine now up to this point. But what about the condition that we call unbalanced, where the first and second vibrations do not necessarily have the same duration? To our major surprise, we found that the model I showed you before would predict that here the performance would be less good than here. That when you increase this duration, the duration of the first vibration, the subjects just get better. But that's not what happens. What happens is a shift in the entire psychometric curve, both in humans and rats. What does this shift mean? In simple words, what the shift means, and I can illustrate what this means, how you get this from psychometric curves, but what the shift means is that the shorter vibration is judged as being smaller sigma, lower intensity, lower something, less. And the longer vibration is more, humans and rats. So for instance, so if the first vibration is shorter, then that first vibration has to be this much stronger than the second in order to be judged as equal. Because if you go 50% and down, you get this value. So it has to be stronger in order to be judged as equal if it's weak, if it's shorter. Well, maybe it's just because of there's some order effect. The first vibration is different from the second, whatever. So then we change the second vibration duration, and we find that the curves switch, they flip. So now the second vibration, if it's shorter, is judged as being weaker, smaller, less intense, whatever it might be. And the second, if it's longer, is judged as being stronger, both humans and rats. And this, as I say, is something that we found that after we found this effect, we went back to what we call the Bible, and we found that it was already written in the earlier chapters in 2005. So we keep going anyway. So this was the first model, right? And it was okay in explaining improvement in the balance condition when two stimuli are equal to each other. But this model fails miserably in the unbalanced condition, because if the first, let's say the stronger one, the green one, if it's a shorter duration than the red one, there's no situation in which the duration makes the relationship flip. You can't cause an inversion of the intensity relationship by duration according to this model. So we made a new model, new hypothesis, that looks like this in the first order, where the percept is not an averaging process, but it's a summating process. And so according to this model, what the sensory system does is it takes each instantaneous value of absolute velocity, of speed, because that's what receptors feel is speed, and summates it to the preceding one. So if it's a summation process, it makes the perceived value increase with duration. And here I show you a linear increase. Of course, it's not linear, but here I show you a linear increase. So how does that explain our result? Because the green stimulus, which is stronger, if it's only for 200 milliseconds, on average would be felt as weaker than the red one, because I just take the green at 200, project it up, and I see that it's on the left side of the red one. So the summation model can allow the relationships to flip according to duration. As I said, it's linear. It's shown linear here, but we know it can't be linear because in biology very few things are linear, but also because the evidence tells us it's not linear, because we know that human and rat subjects don't feel a stimulus that's twice as long as being twice as strong. It doesn't grow that way. So it's not linear. So what we do is we make it exponential, because exponentials work well in biology, as you already heard in the previous talk. So consider this case where you have a 400 millisecond stimulus that's followed either by a 600 or a 200 duration. The two values are equal to each other. So according to the first model, on average, the subjects would say this is equal to this. I mean, of course, 50% of the time they'd be right and 50% wrong. But on average, they say this is equal to this and this is equal to this, because duration doesn't cause a change in the percept. Now, if we take the instantaneous values of velocity, absolute value of velocity actually, and multiply them by a weighting function, and the weighting function is e to the minus t over tau, where tau is some time constant for this exponent, and it says how much each individual value contributes to the percept. So the percept is growing, but not linearly. Linear would mean that the weighting function is constant. It's not growing linearly, but growing exponentially. So we apply it to this vibration, and we take each instantaneous value, multiply it by the weighting function at that time, and then the percept grows over time by this logarithmic function. Then it's stored in memory. So this dashed line is the memory of the first stimulus. Along comes the second stimulus, same sigma, but if the second stimulus lasts longer, it grows for longer. So now the second is judged as being stronger, but if the second lasts less time, it doesn't grow enough, and it's judged as being weaker. So this can explain a lot of things. It can explain the effective duration, and it can even explain why we don't see much change from 400 to 600, because this is a saturating function, and so at a certain point, increases in duration don't really have much effect. So I made a model, but for time reasons, I won't go into in detail, but I think I have pictures in the next slides. So the model is trying to explain a subject's likelihood of judging the second as larger than the first. And what we do is take a lot of data, make a very simple model for how the decision is made based on these variables. And then we actually just fit the model in order to extract the values of these variables. And then using the values of these variables, we predict the subject's performance for a different data set. And the validation of the model is by extracting the parameters from one data set and predicting performance for another data set, can we actually predict what the subject does? And if we can, then there's probably some validity to the model. So these values are the overall efficiency of the observer, the psychometric slope, because some subjects are better than others, and you need to take this into account. So you have a measure of basically how good is the subject. Then you have a bias of the subject, because both humans and rats sometimes have a tendency just to say the second is larger. Some have a tendency to say the first is larger. For rats, it's a bias to turn right or bias to turn left. And that affects the curve, and you have to extract that. And then tau is the integration time. How many samples in history does the subject use weight in order to derive the percept? So this is a decision variable. Then we use signal detection theory. So the choice is based on the actual value of the sigma multiplied by this weighting function. Then we derive d prime, where we take the difference between the means, divide by the standard deviation. That's standard signal detection theory. And the key to this, and I should say Arash did this, and it's very beautiful. The key to this is, this is supposed to be p, not rho. The key to this is that from the d prime, you can actually make a psychometric curve. And I hadn't figured that out, and Arash figured that out, and it's, I think, a very nice insight to make a relationship between the d prime, which is the choice observed for stimulus differences. You can make a psychometric curve. Okay, so these are the results, and for a human and a rat, not the best. I mean, this is typical because it's not fair to show you the best one, but this is typical. And the values from the model, which I can't remember if it's the solid line or the dashed line, one is model and one is observed data, and the model fits it quite nicely and can fit the effects of stimulus duration. So we think we know what are the we think we know how stimulus duration can affect the percept. It leads to an interesting prediction. What if sigma 1 is equal to sigma 2? What if we take trials of that kind and we say, what is the how frequently does the subject judge the first as larger than the second, the second is larger than the first, when they're in fact equal, according to the durations of the stimuli? And it just jumps right out from the model. The model just, it gives you the numbers. You don't really have to do anything. And how do we get the data? It's so time consuming, it's so hard. And Francesca's trained a lot of rats that Ash worked on, it's really hard. And it's so difficult because you have to train the rat on the whole thing, test them on the whole stimulus set, test them on the psychometric curves, on the psychometric data set, and then, lo and behold, in the middle of the psychometric data set are stimulus pairs where sigma 1 equals sigma 2. So if all of these stimuli are of equal likelihood, this is, say, 1 out of 30, I don't know exactly, but it's 1 divided by all of these plus all of these. So every once in a while, there's a pair of stimuli where sigma 1 equals sigma 2. Now, vary the durations of sigma 1 equals sigma 2, and you have to fill it up so that you have enough trials with different durations. So it's a hugely laborious experiment to run. So I'll come to the point of it, so far we only have 1 human and 1 rat, because it's really hard. And so T1, here the durations are equal to each other. Sigma is equal to each other. Durations are equal to each other. Here, the first is longer than the second. Here, the second is longer than the first. And what are the predictions? Predictions are, well, it's very easy. You could say it yourself. If T1 equals T2 and sigma 1 equals sigma 2, on average, the subject should make the choice 50% one, 50% the other. Here, they should judge the first as stronger, and here the second is stronger, and the likelihoods should depend on the relative durations. So the durations go from 100 to 600 for the first stimulus, 100 to 600 for the second stimulus, and then the color is the likelihood of judging the second as stronger than the first. This is the observed data. This is the model. Observed data in 1 rat, the model for the same rat, the model built on another data set. And it's really, for this kind of work, it's really a close fit. And what it says, both in the model and the model predicts, and then we see in the real data, is if the second stimulus is 600, the first is 100, it's very likely that the 70% likely subject will judge the second as stronger. Sorry, the first is 600, the second is 100. Then it's 30% likelihood. And then here it goes up to 70%. And basically, we can predict the likelihood of the subject judging one as stronger than the other based on the durations of the 2 stimuli. Now, what I showed you is a model built with this descending exponential function. But what actually comes out of the model is the integrated value under here. So you would actually get, in everything I've showed you in the model so far, you'd have the exact same result whether the exponential function is decreasing or increasing. The model doesn't distinguish between that because it weights it and integrates it. So the model can't tell you that so far. But we wanted to know that. Of course, we found out that we realized it had already been done. But we wanted to find out for ourselves whether in rats, as in monkeys, the exponential function is in fact decreasing and not increasing. And it's really interesting because lots of cognitive functions give more weight to the last thing that's happened, which is called recency. And it's not, you can't make the assumption that the brain gives more weight to the beginning, which is called primacy, or there could be some combination of recency and primacy. So the way that we did that is we take the vibration and we multiply it by a very gentle envelope so that the mean value of the vibration doesn't change, but either gets very slightly weaker or very slightly stronger. This one would get a little bit weaker, this one get a little bit stronger in time, and in human subjects we show that they can't actually detect the direction. So there's they can't employ some top-down process and say, that vibration's getting weaker, what does that mean? In fact, the slope of this envelope was such that subjects couldn't say if it's getting weaker or stronger. But as you'll see, this thing that they don't know that they're feeling affects their choice. And I think that the predictions are quite clear. If the subject compares this to this, the sigmas are equal to each other, imagine, then if primacy is at work, then this one would feel stronger than this because the early part of the stimulus, which gives the most weight to the percept, is a little bit bigger, and the end of the stimulus, which doesn't contribute as much to the percept, is smaller. Here, when the subject is taking these values and giving the most weight, the values are a little bit diminished. And then when they're giving less perceptual weight, the values are increased. But the summated value by primacy weighting function, this summated value will be larger than this, and by a recency algorithm, this is stronger than this. And the answer is that primacy is at work because stimuli, these are psychometric curves with the different shapes of envelopes, and primacy is at work because when the first stimulus is slightly stronger at the beginning, the subjects judge it as stronger. Humans and rats. So then we have to add some neuroscience to this to complete the triangle, and so we developed a recording system that is better than what was commercially available, made by the technicians in my lab, and this is implanted under surgery. Then the animal wakes up, and there's electrode arrays in one or more places in the cortex. We can put in multiple electrode arrays in different places, and they're movable so that we can study a group of neurons and then move the electrodes and study other neurons. And the rats are very comfortable with this. Okay, so again, what's known from the work of Romo's laboratory, and I won't go into it in much detail now, but what's known is that there's a transformation, a step-by-step transformation of signals within the neocortex, whereby different parts of the different components of the sensory stimulus are extracted and stored and compared in different regions. And by working this out over 15 years or so, the Roma laboratory has been able to specify what kind of operation is occurring in each of these cortical modules. But our starting point is that rats can do this maybe as close to as well as monkeys and as well as some humans. And so this cognitive function, this delayed comparison task on a noisy vibration, is done as well by a very, very tiny brain and with very few functional modules. Okay, how you define the cortical areas, we talked about some this morning, and I don't think there's any perfect answer, but however you divide up the neocortex, there are many more modules in the primate, in the macaque monkey, than there are in the rat. There's no way that this argument cannot be made. We don't know exactly the boundaries, we don't know what's analogous to what, but we know that there are many functional modules here and many fewer here. So the question is, how can the same thing be done by such a small number of modules? And the answer is how do I say, work in progress, under construction. But we do have some insights, and I'll show you a bit of data. one of the insights is that in the rat, so these pink areas are what we're studying now. S1 cortex, I'm sorry for the terminology, it's not stimulus 1, stimulus 2. It's primary sensory cortex, secondary sensory cortex, premotor cortex, hippocampus, medial prefrontal cortex. Is that in the rat, a given region does multiple functions. Premotor cortex is actually very strongly involved in working memory, in the comparison and decision making, all of that. So we see neurons, see neurons that we record at the same time, they're next to each other, that are essentially playing out many different aspects of the same task. They encode the stimuli, they hold the stimuli in memory, they compare the stimuli, they point the rat in the direction of the operation to take. So the strategy that the rat brain uses is to compress many different functions into one cortical module, much more so than in monkeys or presumably in humans. So that works great in this task. Now to get into philosophy, and then I'll show you some data. So what is the cost of this? I mean, if all the same operations can be done with a small brain and with fewer modules, why did we need to evolve a large brain? And I think that the answer that we would find if we could do the right experiments is that there's actually a cost to this. And the cost is that the cognitive functions are not independent of each other. They're mixed together. So let me explain exactly what I mean in the case of rats. So the premotor cortex, which is actually involved with the rat planning its action. In other words, premotor cortex is what the rat uses actually to initiate the action to turn left or turn right. It's also storing information and doing comparison. It's doing a lot of things. So is there a cost to this? I think that there is, because for the rat, I don't think that when they feel the stimuli and they compare the stimuli, I think that they're actually feeling the action that it should take. That it cannot, my guess is that they can't separate what they feel and what they think they should do from actually doing it. So if you take a human or a monkey, we know we can do this with humans. Maybe it could be done with monkeys too. And I'll explain this and then have a look at data and the philosophy. If you take a human and you say, okay, feel these two vibrations and then press a button according to what you feel. They say, okay, I'll do it. And they do it. Then you say, now feel the same vibrations and move your gaze according to what you feel. Fine, they could do that. Say, feel the same vibrations, make the same comparison, now use your foot. So in humans, this could be done with no problem. We're able to perfectly separate what we do from what we feel. In rats, it's very much embedded in the motor system. So we've tried this some, we try to teach them to do a different action according to the same stimulus. And it's very, very hard because the action that they take is very much embedded in the entire decision-making process. So we can't delegate a motor system to do many possible things according to the percept. So I think that one of the costs of this, one of the costs is that functions are not as well separated.
So I think the rats, the premotor cortex of rats and humans and monkeys is very similar. In the sense that perhaps the most interesting observation to us is that premotor network, which was considered premotor, for preparing for motion, voluntary action, is that depending on the task component, you can see
all many
the
different
working memory
phases.
component, the comparison process,
Yes.
and all the process associated to the task.
Yes.
And it's very redundant. It's widely distributed in premotor network plus prefrontal cortex. So in a sense, I think perhaps the rat has less structures, like the monkey, die by sure. But at the end, the premotor network contains exactly the same properties
Yeah, yeah.
as in
Yeah.
rat as in monkeys.
Yeah.
I think.
Right. So I know that that when you study these many different regions, we're familiar with your findings that a working memory neuron could be found in many.
Yes.
So if you look at an individual neuron in your data, it would be very hard for you to say which region it came from.
Absolutely.
If you look at one neuron.
Yes.
So there's much overlapping of functions. I agree. Okay, so this I just put in this morning from due to Henry's talk, because barrel cortex is connected to prefrontal cortex, which is on the medial to premotor cortex in the rat, but it's a very weak connection, and so but it's probably an important connection. So how can a very weak connection actually transmit information? And that's the question raised, I think, by the lecture this morning. And one way that we think the information is transmitted is by coherence in local field potentials, which makes the sending neurons send a message at the same time that the receiving neurons are receptive to it. That's what local field potential coherence can do. And there have been studies about this between hippocampus and prefrontal cortex in rats. I kind of didn't believe it because I thought it was something that people thought would look good, so they look for it and they find it. And it became sort of very fashionable to find coherent oscillations, and so I didn't believe it until I saw it in our own data, and now I believe it. And what we see is that between prefrontal cortex and sensory cortex, again, sorry for the terminology problems, this is stimulus 1, stimulus 2, and the recordings are between sensory cortex, barrel cortex, whisker cortex, and prefrontal cortex. Just before when the rat goes into the nose poke, and he knows that the stimulus is about to begin, they suddenly become very coherent in 8 hertz, in theta rhythm, local field potential. And then remain coherent for at least the first few hundred milliseconds of the first stimulus, and then the coherence really drops off when the stimulus ends. Which, if you want to sort of attribute a function to this, it makes sense because prefrontal cortex doesn't want anything coming from the sensory system during the delay because it might disturb the network. And so the coherence is mostly shut off, and then again begins a little bit during the second stimulus. So we're working on this, and it looks like there's actually local field potential oscillations are ways to kind of to our surprise, because we were skeptical about it, but our evidence is that it actually is consistent with facilitating communication between regions. And then this is the baseline. This is the distribution of phase relationships between sensory cortex and prefrontal cortex during baseline when the rat is just doing other things. And then just as it goes into nose poke and it's just waiting for the stimulus to begin, they suddenly become very compact in the phase relationship and remain so during the first stimulus and then a little bit less in the second stimulus.
Does the coherence in any way correlate to the duration of your stimulus?
The coherence is strongest during the pre-stimulus period and then during the first couple of 100 milliseconds. But
independent of the total duration of your...
Well, we have so many variables. In this particular experiment, we didn't vary stimulus duration. Okay, so let's have a look at a neuron from sensory cortex, primary sensory cortex, barrel cortex. And this is one neuron. This is all neurons put together. So something like 200, probably more than 200 neurons put together. And this is a histogram of responsiveness over time, averaged across all the trials. And it happens it's for 600 and 400 millisecond stimuli. You can see that as the rat is going into the nose poke, there's lots and lots of activity. It's whisking, it's touching a lot of things. And then it settles down to a low rate in expectation of the stimulus. And the stimulus begins, the firing rate goes up, drops down a little bit, interstimulus delay, and second stimulus. So what is the signal that's being given by these neurons? If we look at one vibration, here's the vibration, it's converted to speed, it's from velocity, it's converted to speed. And so this is one vibration which is given at 3 different sigma values. So the in velocity space, the vibration is stretched to change the value of sigma. And this is the response of the neuron in spikes per second for the same vibration, but stretched and compressed. And you can see that there's kind of this, it's not a continuous firing rate, but it's one that varies from moment to moment within the vibration. And for different values of sigma, it kind of varies in the same way. Why does it vary? Well, if you align the trials, this is the exact same vibration repeated 60 times. And you can see that in different trials, the action potentials, the spikes, occur at kind of the same time. And then if you reduce the vibration or increase it, then within one value of the vibration, the spikes are again aligned, but of course, stretching and compressing it changes the overall firing rate. So in the sigma, so how do we actually find what makes a neuron fire? The most direct method is to do some form of spike-triggered averaging, which means you take each occurrence of an action potential and you say, well, what was the stimulus just before that? What happened that made that neuron fire? And so here I'll show you, it's a movie that shows the stimulus moving in time. Each dot is an action potential. And then here I'll show you the average over all preceding windows of the stimulus prior to each action potential. And you'll see this spike-triggered average building up. This is a 1 second stimulus, just to map out the neuron. It's 1 second. Okay, so here's for the first set of action potentials, this was the average stimulus before. And then we add more action potentials, so we keep averaging this preceding window. And now we're about 300 milliseconds in, and you can actually start to begin to see the spike-triggered average. This is being done by Amir Judaki. And now the filter or the kernel of the neuron is actually becoming quite clear, which is amazing to us. In 1 second of data, we know the filter of this neuron. And then we have to do lots of statistics and so on. But in 1 second, we can see what it is that the neuron is reporting. And what is the neuron reporting? It's not reporting sigma. It's reporting, because sigma is based on a distribution of values, right? Sigma is not an instantaneous event. Sigma is something that is accumulated over time. What the neuron is reporting is what happened in the last 10 or 20 milliseconds. So in that triangle that I showed you, notice the distinction between encoding, the sensory coding, putting the physical stimulus into a spike train. That's what this neuron is doing. It's not decoding, though, because the neuron is not saying what is the overall value of the stimulus, which is what's necessary for the task. It's saying what are the tiny elements that make up the stimulus. But in order for the animal to do the task, it has to decode all the spikes that come from sensory cortex. So these are different ways that a stimulus the firing could be decoded. It could be decoded as the cumulative mean firing rate. It could be decoded just by summating the spikes, or it could be decoded by weighting the spikes and summating, which is how we think it's decoded.
But the cell we just looked at was in the somatosensory cortex,
Yeah,
right?
right.
So we wanted to do this.
We wanted to do this. So it's what we hoped it would do, and that's what it does.
Exactly.
And I should say this is for 45 to 50% of neurons, have very clear, very what we call local filters. The filter, what the neuron tells the rest of the brain is what's happened in the last 10 or 20 milliseconds. And that's true for about 50% of neurons. For about 50% of neurons, we don't see a really strong relationship to stimulus. Okay, so we can say, okay, the rest of the brain is getting the spike train, trying to figure it out and make a choice. So how is it making a choice? How is the rest of the brain using this sequence of action potentials? And this shows ways that it could be used. Then we can say if the brain is using the spike train according to these potential models, and I forgot to put this is the primacy weighted, I forgot to put the recency, but it would just look the other way. It would look reversed. What would be the choice, what would be the behavior of the animal? So to do that, we make a neurometric function, which means taking the spike train, making a choice from the neuron's activity based on what happened in one trial, doing that for many trials, and then trying to guess the behavior that the rat would have according to the method that we're reading the spike train. And so we get a neurometric function, which is what an observer would choose from the spike train according to how it's reading the spike train. And here, from one rat, these are the red dots are the actual choices of the animal for two different stimulus duration comparisons. And these, again, are data that are kind of hard to get, so we don't have, it looks a little bit disorderly, but it's one rat, one session, because that's how long you can keep a neuron. And the solid lines are the expected choice of the animal if the sensory cortical neuron is read or decoded according to different models. And if it's decoded according to our preferred model, the weighted summation primacy model, then the observer, this solid line, would actually be pretty close to the actual animal. But if the observer is using any of the other possible models, then the choices of the observer would be quite far from the actual choices of the animal because the solid lines are not close to the dots. So what this means is that sensory cortex is encoding the stimulus, but something has to be done to the signal from the sensory cortex to explain the choice of the animal. So we think that the weighted summation model, which we've seen as a good is a good model for behavior, but it's being done on what the rest of the brain gets from sensory cortex. Sensory cortex has not yet done the weighted summation. In premotor cortex, these are a lot of neurons put together. Neurons fire during the stimulus, during both stimuli, during the delay, but they do very, very different things, and this is what we were already talking about before. So these are two neurons from premotor cortex of the rat, and this is, I would label it kind of as a working memory neuron, because the firing rate during the delay, and especially during the late part of the interstimulus delay, the firing rate is very well correlated to the value of the first stimulus. But in fact, what we've shown is the relationship of the firing rate to the perceived value of the sigma. That is what, from the rat's behavior, what we think it's feeling. And the relationship for premotor neurons, for almost all the premotor neurons that we've studied, the relationship of the firing rate is stronger if we look at firing rate in relation to what we think is the stimulus, is the perceived stimulus, than the relationship between firing rate and the actual value of sigma. So the firing of these neurons is related to what the animal feels even after it has, once it has already weighted the stimulus according to time. Does that make sense?
How do you define the perceived?
Because from the animal's behavior, we know how a given stimulus should be summated. We apply a weighted summation to this vibration, and we say, okay, according to the weighted summation, these are the values of the different stimuli. If we don't weight it by time, so essentially what weighted summation does is make stimuli that have different durations have different values. Without weighted summation, a sigma equals 50, let's say, will have the same value whether it's 100 milliseconds or 400 or 600. This is another neuron in premotor cortex, recorded at the same time. It may be even from the same electrode, so they're very much mixed together. This neuron encodes the first stimulus, and then during the second stimulus, it separates according to the choice of the animal. So the bottom line is that in sensory cortex, we see a very local code. And in premotor cortex, we see many different operations that the brain has to do. We see them folded in next to each other, which is also true in primate.
But I don't, maybe I'm slow, but so the point you're making is that in the waiting period, we have a ramping function now on the response of the neuron that relates to the inferred sigma by the animal.
Yes.
Right?
Yes.
But now, how is that mapped to the decision time? So
in this neuron or the other neuron?
Right, exactly. How should I map the waiting period to now the decision period? How should I
so a neuron like this somehow has to be transmitting a signal to other neurons. This neuron...
This neuron... Not the same neuron.
No, it's two different neurons.
Okay.
This is what we call a working memory neuron, and there are lots of these, which carry a lot of information about what the preceding stimulus was, especially the percept of the preceding stimulus, not so much about what the rat is going to do with the stimulus. Here it's separated according to choice. The color is the choice. This is a neuron that doesn't seem to say too much about the preceding stimulus, but it says a lot about what the rat is going to do with the choice. So two different neurons.
What Matthew wants to say is that you have a micro brain there that has recreate all the task components, the sensory input, the work and memory component of the task, and the decision report.
Yeah.
Right.
Yeah.
Okay.
And mixed together in very funny ways, because this comparison neuron actually encodes the first stimulus. So you could almost say this is like a sensory neuron, right?
Yeah.
Doesn't have much memory, but has a lot of decision making. And then this neuron doesn't have much sensory coding. Actually, here's this, during the first stimulus, no sensory coding. So it must be getting the memory trace from other neurons because it's not getting it from itself. So these are very interwoven in very complex ways.
And this neuron in the working memory neuron is in premotor cortex.
These are premotor neurons.
And the comparison?
Both are in premotor cortex. So we have neurons from lots of other regions, but I'm not putting down showing all the data.
Given that it's a recording from premotor cortex, do you think that some of that activity might be related to a motor element? There's a element of motor preparation.
Yeah, for sure. Yeah.
So there's this issue about how you treat working memory is in the cognitive literature. When you in the human literature, you think about working memory as representing something in a very abstract sense.
Okay. Yeah.
Some kind of thing that prefrontal cortex would be doing in the human brain.
Yeah.
So, is there a way of teasing apart sort of the slightly more cognitive side from the more motor side that we'd see?
You were going to answer that?
Well,
I thought you were going to say something about that.
In the monkeys, for this kind of cells, this kind of activity can be treated as expectation signal, for example, for the second stimulus. However, you see that the responses are a function of the first stimulus, whatever is the quantity in your case, or our case for the frequency. But in some controlled tests, we have delivered exactly the same stimuli and instructed the animal to make the same push button presses. And in a large number of the neurons do not show any more of this response. So you take apart the motor component or the expectation signal.
Okay.
For that,
And
for
that's
certain number of cells.
That's still from premotor cortex?
Yes.
Okay.
Yeah, what he said.
Okay.
So we're aware of that control, and we would like to do it, but we haven't done it yet. That is to separate the action from all the previous cognition. And another interesting approach that we probably can do if we get enough sessions is to actually extend the post-stimulus delay. That's kind of interesting because if the neuron's activity is more strongly connected to the actual motor output, then it should extend through the entire post-stimulus delay. But if the neuron contributes to choice and then gets out of the circuit, then during the post-stimulus delay it should decrease. But and what we've seen in a lot of these neurons, but it's not a proof, but a lot of them for 200-300 milliseconds in the post-stimulus delay, they maintain the choice selectivity.
And 0 here in the decision, the last panel, 0 is the movement onset.
Yeah. Is where here? This
Yeah,
0?
The green and black plot to the
Oh.
right. 0 there is the movement onset, or the
No,
stimulus
no.
offset, or stimulus
No,
offset.
stimulus offset is 0, yeah.
Okay.
Yeah, and these are statistical significance. These and these.
All right.
And these. Yeah.
Cool.
Why is it that you have used sigma for time from S1 and perceived sigma from time from S2? So in the
Yeah. Oh, because there is no perceived sigma because you can't group these together because they have different durations. So during S1, during the first stimulus, it's simpler graphically to plot sigma, because if you plot perceived sigma, then it changes during the course of the stimulus. And so this stimulus lasts longer than this one. So you have two different perceived, so you have the same perceived sigma up to this time. Then this set of stimuli has stopped, this set of stimuli continues. So you have two different perceived values. So it's just, it could be done, it's just much more complicated to find a way to plot perceived sigma during the first stimulus, just graphical illustration reasons. But once the stimulus is ended, right, during the delay the stimulus is ended, so any given stimulus can be mapped onto a perceived sigma. But during the stimulus it's very hard to map it onto perceived value because it's changing according to duration. Okay, so the last part, the last part is very, very quickly, it just takes 2 or 3 minutes to explain this. So what we've shown, the main message is that differently from what we had expected, so we found some things we expected, some we didn't. We found that for stochastic stimulus, the human and rat observers get better with stimulus duration up to about 400 or 500 milliseconds. Unexpectedly, the duration of the stimulus produced a bias in so the perceived value was not stationary over time, but grew over time. And I showed you that a primacy exponential weighted summation really accounts for behavior very, very well. So that's the main thing that I've shown you. So we thought that if the duration of a stimulus plays out on perceived intensity, maybe it would work the other way, so that the intensity of a stimulus would have an effect on perceived time. And we found that it did, that we find that it does. And to me this really touches something very, very deep in systems neuroscience, because all of us who study neurons, we record neurons, we plot the data of neurons, what is the y-axis in every plot? 95% of the plots that we make, what is the y-axis? Rate, which is? Spikes per second, spikes per whatever, spikes per bin, spikes per unit of time. So if that's the coding mechanism, it's based on the assumption that the brain knows the time, right? That it works perfectly if the brain has a perfect measure of over what time span it should collect the spikes. But what if that's not true? How can we separate the perception of a stimulus from the perception of time if the brain doesn't know exactly what time is? So what we find, and I'll show you now, what we find is that time and perceived stimulus value they're inseparable. You just can't do one without the other. You can't measure time independently of the properties of the stimulus, just as you can't measure the properties of the stimulus independently of time, because the brain uses time to compute things, but it doesn't exactly know time. So my student, Alessandro Toso, he's a medical student, but now he's starting a PhD, and he gave the exact same stimuli. You don't even have to change the stimuli, but you just change you change what you ask the subject. So he gave the same kind of stimuli, which vary according to the sigma, and they vary according to the duration, but now instead of saying, tell me the sigma difference, you say, tell me the time difference, the duration difference between the stimuli. So T2 versus T1, tell me if T1 is larger than T2 or T2 is larger than T1. And you do that for stimuli which can have different relationships in sigma. And the answer in 12 healthy subjects, as we had expected, is that the relationship, the perceived durations is affected by the strength of the stimulus. And just to decode these curves for you, what it means is that the stronger stimulus feels longer. It feels like the duration is longer. So stronger is longer, weaker is shorter. So you cannot separate time from the properties of the stimulus. And the effect is exactly symmetrical to the effective stimulus duration on perceived intensity. So when he was collecting these subjects, one was completely different from the others. And so we went back and looked at the questionnaire that we have from all the subjects, a few pages of data that we're required to collect on all subjects. And we found out that this guy who's completely different from the others is healthy, has no kind of symptoms of any kind, but it turns out from the data that we collected that his brother is under treatment for schizophrenia. So in medical terminology, this guy is considered called at risk, meaning no symptoms, but probabilistically there's some increased likelihood he's at risk for developing schizophrenia at some future time based on something that's happening based on family, the family genome. So perfectly normal, but at risk. So my student Alessandro, he was a medical student. He's going to go into psychiatry after his PhD, but he's really interested in sort of the healthy and the not so healthy brain. So he found 3 more at risk, and the result is that they have a massive effect of duration of stimulus intensity on time judgment. So they're really not able to separate, even normal healthy subjects are unable to separate intensity from duration, but the at-risk subjects who have no symptoms, so this is like a very sensitive marker for something that's different in the brain, they have much more difficulty, much more confusion between the stimulus properties and the duration of the stimulus. So then we're going back to rats, and this is the grid of stimuli for a rat that's doing a time difference task now. And they're still learning, but this is quite promising that we can get rats to do the time difference task. So thank you for your attention.
Thank you. All right, fantastic talk. It's question time. So one thing that I found interesting, you haven't really talked about the reaction times at all. Well, usually, sort of from a psychophysical perspective, accuracy and reaction time are often tightly coupled.
Yeah.
And certainly, we see these strange reversals in accuracy that you are inducing.
Yeah.
You would predict that reaction time should follow exactly that same regularity.
Yeah.
So, have you looked at that? Do you know anything about?
Yeah, we're interested in reaction time. We have different behavioral tasks in the lab where we get a lot more reaction time data than this task. This task, we had a trade-off between measuring reaction time and controlling stimulus duration, because in a lot of monkey studies on motion perception, where you have these moving dots, the monkey is allowed to make its choice as soon as it wants to. So then the monkey has a trade-off between collecting the data the sensory data for longer and being more accurate, or getting the reward or hoping to get the reward earlier. And what's been found is that when the dots have more coherence, so the task is easier, the monkey reacts more quickly because it gets the necessary evidence more quickly and makes its choice. And when the dots are less coherent, the monkey waits longer. So we could have done that in our experiment, let the rat go when he wants to go, but rats have a very strong, in the accuracy, in the speed versus accuracy competition, rats give a lot of weight to speed. they're impatient and they want to go fast. So if we allowed rats to do the sigma comparison and to go when they had enough data, they would really go very early and make a lot of mistakes. And it would also make it very hard to do this measure of integration over time, because they would probably go, I mean, they would be very compressed in the time that they would wait. So that's why we decided to make them stay for the entire stimulus, because it's the only way that we can make sure that they're actually receiving the entire stimulus. Otherwise, they'd go early. So they wait for the entire stimulus, and then they wait for the go cue. So the only reaction time that's available is the time from the go cue to the spout, and that's really fast. They go very fast, whether they're certain or not certain. There's some small variation. They go a little bit faster when the stimulus is stronger and when they're more certain, and correct trials are faster than incorrect trials. But there's not that much room to play with, because it's a very stereotyped motor task. There's another task where we have lots, where there's well, I won't go into it, but we can get reaction time data quite well for another task.
Yeah, so Matthew, Henry ended his talk with a rather controversial statement that brains aren't machines. Your talk beautifully illustrates how what we might have thought as a complicated decision-making process plays out in the brain of an animal, and although we don't fully understand it, we now have a grasp of many of the major components of the system, of a mental apparatus that detects stimulus properties, compares them, stores them over time, and enacts a choice, which is played out through the motor system. From your point of view, how do you see this analogy with, I mean, you also have a nice mathematical model for this. How do you see the analogy between what's going on in the brain and a machine, for instance, like we might imagine building a computer or a robot that could do this task? What would be missing in the robot compared to the rat?
I think that we're so far from being able to work out what's happening in the brain in this task in a mechanistic way, deterministic way. We're so far that I wouldn't even think of a machine doing it. For instance, what I showed you in neuronal activity, I showed you some neurons that do some things. I think that we could say in a mechanistic way, no, I'm quite sure that we could say in a mechanistic way, together with, putting our ideas together with many different laboratories like Carl Svoboda and Rasmus Peterson and so on, we could say what makes a neuron in the sensory cortex fire the way that it does. That could be done pretty much mechanistically. But if you look at our data, we've seen neurons that look like they have a working memory. They're models for working memory. No one knows how a memory is stored. And I showed what is the simplest possible way that we can envision a memory, which is the firing rate of a neuron in relation to the information that's stored. But there's so many other ways that memories could be stored. There are many different population mechanisms that I didn't look at all. There are things that you can't even see in spiking, because there's the most appealing theory for memory, the Sodek's Omri Barak model, is that it's actually stored in synaptic weights that you don't even see in spiking. So I've shown you what looks like a working memory, but I don't know, nobody knows how a memory is stored. So I think that you say, okay, if we could make a machine that does this, we're so far from that.
But I think you're being too modest, because you have a model which, by your own statement, comes close to reproducing the data.
No,
of course, we don't know.
Simulates the data.
We don't know the full details of how the brain is doing this, and we never will. But in terms of building an artificial device that could replicate the behavior, we are surely, once we have your model, not too far off doing that. And
you mean the weighted summation provides a simulation of the behavior? I mean, we can tweak the model so that the model can predict how the rat will behave, but it's not an explanation. It's a replication. It's a simulation of the data.
So I was just wondering the details about the training. So when you mix both the different sigmas and different durations, were they mixed from the very beginning of the training, or
Yeah.
you train animals and subjects
Yeah,
at
it's
the
a
same
super
duration?
fantastic question. And the reason that it's a fantastic question is someone could say, okay, if they're trained only with one value of duration, then the answer mathematically for the brain is equivalent to in averaging and summating. Both of them give an equally correct answer. So maybe rats just prefer to summate rather than average. It works. Then if they've found this computation that works, then you give them different durations, and now their summation algorithm confuses them. And so that's one possibility. So the first rats we did train with fixed duration, but then when we found out in those rats that changing duration affects the position of the psychometric curve, we thought, okay, we better train them with many different durations so that they can't settle on the summation algorithm just because it works for those. So now we train rats with varying sigmas and varying durations, and there's no difference at all. What I can say is that the effect of time on the perceived value of the stimulus, it's not at all affected by your knowledge or by your training. And the way that it's deeper in the brain than that. It's more primitive than that. And the way that I know that is that we test ourselves a lot. And once the people working on the project once we've realized the effect of time, we've tested ourselves. We don't include the data, of course, in the manuscript because that would be ridiculous. But just for our own knowledge, we test ourselves. And even if you know that a longer stimulus makes it feel more intense, so you say, okay, so knowing that I'm going to decrease my estimate a little bit for this and then increase it a little bit for this, you can't come close to eliminating the effect. In fact, one person in the lab who understood these data, we retested him with different durations and everything was switched around because he was trying to compensate, but he way overcompensated. So he said, okay, this was a long stimulus, so I'm going to make it judge as being very, very weak because I know that the increased duration made it feel stronger, so I'm going to compensate for that. And you can't do it. You can't do it. You can't take away, you can't use your knowledge of duration and normalize intensity according to your knowledge. It's just what you feel.
And just one more question. Do you think, is there anything special about somatosensory?
No.
So you think this could transfer to auditory?
Well, transfer is a different question, but does it work in other modalities? Absolutely. I mean, we do this, we just made it into a sound, and in 10 seconds we showed, okay, that's an exaggeration, in 10 minutes we proved that there's the duration effect for acoustic stimuli, and we also proved in 10 minutes that it's by the envelope, we proved that it's a primacy weighted function, not a recency weighted function. We proved that longer stimuli feel stronger. In auditory stimuli, the effect comes out in the first 20 trials. Very, very clear. And sorry, we didn't do vision, but we looked in literature, and there's been some studies with vision. When there's moving dots, people have to judge the duration, and the more dots there are, the more confusion, the longer it feels.
So, do rats or humans develop a prior expectation of which is the velocity of the vibration by training?
No, well, they might, but they shouldn't, because there's nothing in the stimulus set that is predictive. There's you mean before the first stimulus or before the second?
I mean that, is there anything indicating that they have a prior expectation before the stimulus?
Okay, yeah, that's another super fantastic question, and I could talk a half hour about it. But so ideally they shouldn't, because there's nothing that they can predict. So the perfect machine doing the task would not make any prediction because it can't. the brain does make predictions. Humans do it, rats do it, and it's called the prior. And the prior has a very strong effect on working memory. Ranulfo is working on that. We're working on that. We collaborate with some of the same theoreticians. Jonathan Loewenstein, he's working with Ranulfo, he's working with us, and he's the world expert on the effects of priors. And here's the way it works. When you have the first stimulus and you put it into memory, over the duration of the delay, the memory of that stimulus, the value of the memory is affected by the previous stimuli. And the tendency of subjects, humans and rats, is to move the memory towards the prior, towards the expected value, towards the mean.
So thank you for the talking. I want to speculate a little bit because all the stimuli you showed us is somehow we could call they are acoustics, okay, stimuli. Yeah. Vibration is acoustic, sound is acoustic, ultrasound is acoustic as well.
Okay.
Okay, we know that rats, they have used to communicate in high frequency.
Mm-hmm.
And the human doesn't. We don't use high frequency to communicate.
Okay.
So that means what I'm speculating that is our let's say our response for subsonic frequency or less connected to communication, because the way rats use for communicating high frequencies, also they are using the same device. You know what I mean? They're using the
whiskers for high frequency.
Yes. Or not, because high frequencies are very directional. That means maybe I'm wrong, but they would also use. There's a complementary between the auditorial.
What is the evidence that they use whiskers for acoustic?
No, I don't have evidence. I'm speculating only. I'm sorry.
Okay.
That means they would use. So in that sense, that there's the premotor, let's say, answer they have for the sound, the low frequency sounds, would be also connected to the high frequency sounds, because they had to do actions. And this is completely different in humans. We don't have to communicate in high frequency and even in low frequency. You know what I mean?
That's what you're saying, is that there's a bias in the sensory apparatus.
Yeah, that's what I'm saying. And also, if you modulate vibration, you are modulating the speed, okay? So by modulating the speed, you are modulating the acceleration. And acceleration means force, okay, in physical terms. So that means also there is forces conveying also actions. And also for the rats, but not for the humans, what I mean. Yeah?
So the vibration is ultimately converted to force, and the receptors in the skin, which means in the whisker follicle for the rats, are probably force receptors. That's probably what mechanoreceptors respond to, and that's true also in humans. So it's possible to make, and we've made it together with, I don't know if this is, with the applied math department at my university, models for whiskers and the force that they transmit into the follicle, which is converted into action potentials. That's probably what receptors do. So it's true that they do receive force. That's true, but in the end, I don't know. It becomes a sensation. I'm not sure where to go from that. From there, rats probably do. No one has proven it, but our guess, and we talked about this with Tony 4 or 5 years ago, is that rats may very well use whiskers for acoustic sensation of some kind, but low frequency. By low, I mean under 120, 130 hertz. The reason is because those are the resonance frequencies of the whiskers. The whiskers don't vibrate if you put them in the presence of a high frequency vibration. They don't resonate. If the air pressure is moving at 100 hertz, then there are some whiskers that may be taken up by that and may resonate. No one has shown this, but I think it's a good hypothesis, and I think some students somewhere should do a PhD thesis on this, and I think they'd get good results. So rats could hold their whiskers out and then feel for vibrations that come through the air, not through an object, but through the air, but not high frequency.
All right, Matthew,
Thank you.
Thank you very much.
Thank you.